Hi, this is Dr. Anjali here. We'll deal with common anatomy questions. Fox-Fordyce disease is a disease of eccrine glands, apocrine glands, sebaceous glands, or pilosebaceous glands. It's a straightforward question. It is apocrine gland disease. So it's also known as apocrine miliaria. Okay, these are itchy bumps which are skin colored, and they're seen around the hair follicles, in the underarm, so the axilla, the genitalia, pubic region, and around the nipples, okay, the periareolar region. This is where the apocrine sweat glands are found usually. So it's basically a blockage of the apocrine sweat gland ducts, okay, causing these skin-colored bumps. There's another term, Fordyce spots. Okay, these are ectopic sebaceous glands, that is the oil glands. They're not associated with hair follicles, and these are usually present on the oral or genital mucosa. So it's at the border of the lip, the vermilion, the insides of the cheeks, the buccal mucosa. It can be seen on the glands as well as the shaft of the penis, and on the vulva and vaginal mucosal margins. So you can see here the small yellowish dots. Those are just ectopic sebaceous glands. There are some other ectopic sebaceous glands with specific names like Montgomery glands, that is the ones on the areola of the breast, and the Tyson glands, which are the ones on the penis. Miliaria is a disorder of sebaceous glands, apocrine glands, eccrine glands, or holocrine glands. So miliaria is the sweat glands getting blocked, and those are of the eccrine sweat glands. Okay, so sweat glands of two types, eccrine and apocrine. Eccrine are the ones that are usually present all over the body, that are to deal with thermoregulation basically. So they are the they exude sweat, whereas apocrine, as we just discussed, were present in the axilla and genitalia and mammary. So they're not really important for thermoregulation, and these are always connected with the hair follicle. Holocrine glands, actually these are sebaceous glands. So holocrine is when the cell gets decapitated. So that is found at pilosebaceous units or hair follicles. Okay, those sebaceous glands are holocrine glands. Melanocytes are present in the stratum corneum, stratum basale, stratum granulosum, or the dermis. Melanocytes are present only in the stratum basale normally. So here's a diagram of the epidermis. So the uppermost layer is the dead layer, the stratum corneum, right? The flattened cornified cells, keratinocytes. Below stratum corneum is the granular layer, and then the spinous layer, which is the thickest, and a single row of cells forms the basal layer, which has mainly keratinocytes interspersed with melanocytes. Below that is the basement membrane and the dermis. So here's a more detailed diagram of the epidermis. The uppermost layer is the stratum corneum. So from the basal layer to the stratum corneum, you can see the cells are becoming more compact and flattened out. So on top of the stratum corneum, this flattening is due to the presence of the filaggrin, that is filament aggregating proteins. And filaggrin eventually disintegrates to form the natural moisturizing factor. That keeps the cells together, okay, and that protects, that retains moisture within the epidermis. So when there's a mutation in the filaggrin gene, filaggrin producing gene, you get ichthyosis, and you also do get, when there are mutations, you will get atopic dermatitis and other sorts of systemic allergies, right? The second layer is the granular layer, where you have a lot of keratohyalin granules present in the cytoplasm of the cells. And below that you have the spinous layer, which again has granules present in the cytoplasm. These are called Odland bodies. Now Odland bodies release lipids into the intercellular space in the epidermis, and these have an epidermal barrier function, and they also make sure intercellular connections are kept together, right? So the spinous layer is the richest layer in desmosomes. Desmosomes are intercellular connections in the epidermis, the cell-to-cell connections in the epidermis. And in the basal layer, hemidesmosomes connect the basal layer to the basement membrane zone. Okay. So the epidermis is formed mainly of keratinocytes. You also have melanocytes in the basal layer, Langerhans cells within the epidermis, and Merkel cells also in the basal layer. So the cellular progression from the basal to the skin surface is 28 to 30 days. The brick-like shape of the keratinocytes is provided by the cytoskeleton, which has keratin intermediate filaments. And the stratum corneum, you have to know, has by the time it's reached the top, it's lost the nuclei and cytoplasmic organelles. So what are melanocytes? Dendritic cells, they're present in the basal layer of the epidermis. These distribute melanin in melanosomes to the adjacent or to the connected keratinocytes. So one melanocyte will be supplying several keratinocytes. Okay? These are derived from the neural crest, very important point. And the number of melanocytes does not really differ between different skin types. So whether fair or a darker skin type, number of melanocytes may be the same. It's the nature of the melanin in the melanocytes and the size of the melanosomes which accounts for the difference in the final skin appearance, as you can see here in the diagram. Okay. Langerhans cells are also dendritic cells, and these are mesenchymal origin, so they derive from the bone marrow. These are antigen-presenting cells, so they present the antigens from the skin to the local lymph nodes. So they play an important role in adaptive immune response. Merkel cells are derived from keratinocytes, and they are also present in the stratum basale. They are highly aggregated on parts of the skin that are used for touch. So they are basically sensory receptors responding to touch. So in the dermis is composed of these extracellular matrix proteins embedded in a ground substance. So the main proteins are collagen, which is about 80% of your dry weight of the dermis, and elastic fibers, which is just about 15%. Water actually constitutes about 60% of total weight of the dermis. That's because the ground substance comprising of glycosaminoglycans and proteoglycans, these complex sugars, they have a high water binding capacity. Okay, quick embryology. You just need to know hair follicles start to be seen from 9 weeks intrauterine life, as well as nails as well. The first hair follicles are seen around the eyebrow and the chin. Sweat glands start from week 9 and they go up to week 15 to 16. Sebaceous glands develop around week 15. Langerhans cells around 12 weeks. Melanocytes can be seen around the 4th month, and elastin fibers around 22 weeks. Okay, coming to hair basic anatomy. Growth phase of the hair is anagen, telogen, catagen, and metagen. This is a very straightforward question. Anagen, of course. From this diagram, you just need to know that the anagen phase is the growing phase, as you can see. It's the longest. It can last from 2 to 6 years. Okay, this depends on the individual. And the catagen is the regression phase. So the bulb, as you can see, has contracted, and catagen is a short-lasting phase. Then it goes into telogen, which is the resting phase. And telogen is usually around 3 months duration. After telogen, the hair is then shed, and then the new anagen phase starts. Sebaceous gland activity is mainly controlled by which hormone? Estrogen, androgen, progesterone, or cortisol? So sebaceous gland activity, again, the pilosebaceous unit is is the basis for acne, right? And that is controlled by testosterone. So androgens was the answer. Merkel cell is derived from ectoderm, neural crest, mesoderm, or endoderm. So I said Merkel cells are derived from keratinocytes. Keratinocytes are derived from the ectoderm. Melanocytes are derived from the neural crest. Okay, and Langerhans cells are the mesenchymal cells. Most abundant amino acid in collagen is proline, lysine, threonine, or glycine. The answer is glycine. What does lines of Blaschko represent? Is it lines along the lymphatic channels, lines along blood vessels, lines along nerves, or lines of development? So it is actually lines of development. So lines along nerves, it's not exactly lines along nerves, but dermatomes would be divisions along each spinal branch of the nerves, right, on the body. So Blaschko's lines are invisible lines, but they are basically, they represent the migratory pathway of the epidermal cells in the development of the fetus, right, from originum to their final destination. So certain skin conditions like linear epidermal verrucous nevus will show up along Blaschko's lines. Epidermal turnover time is 14, 28, 40 or 60 days. So I've already told you this, it's around 28 days. Weakest portion of the DEJ, dermoepidermal junction, is stratum basale, lamina densa, lamina lucida or lamina fibroreticularis. The answer here is lamina lucida. I'll show you a diagram of this. Collagen 7 is present in which layer of basement membrane zone? Hemidesmosome, salt split layer, lamina densa or sublamina densa. The answer here is sublamina densa. So again, you can just look at the diagram and have a basic understanding, and then you'll remember these points. So this is the basal cell layer, and it's marked the basal keratinocytes as well as the hemidesmosome connecting the basal keratinocyte to the basement membrane zone, of which there is a lamina lucida, which is actually electron clear zone. So it's easy to remember. Lucida means clear, so it's not the strongest layer in the basement membrane zone. Simple. Lamina densa, to which the anchoring fibrils of type 7 collagen are attached, and type 7 collagen is present in the sublamina densa. Okay, that's the hemidesmosome. Well, on top you can see the desmosome connecting two epidermal cells. Keratohyaline granules are present in which layer? Stratum corneum, spinosum, granulosum or basal. So easy to remember, keratohyaline granules and stratum granulosum. Function of Odlin bodies is antigen presenting cell, epidermal barrier mechanism, filament aggregation, or none of the above. So Odlin bodies, I said, were present in the stratum spinosum. These release lipids and they have a role in epidermal barrier mechanism primarily.